My name is Dr. P.C. Patel, Professor and Chief Soil Health Specialist, Anand Agriculture University and Parul University, and Mati Agromart Private Limited, Anand, Gujarat. So the topic of my YouTube is role of iron, zinc, manganese, and copper in agriculture production. So, as food grain production increases with time, the number of elements become deficient in soil, and crop also increase due to imbalanced fertilization use. So, at present, we are observing the multi-nutrient deficiency in soil due to indiscriminate use of chemical fertilizer, pesticide, fungicide on plant and in soil. So resulted in there will be imbalanced nutrition in soil, and you know that there will be the synergistic and antagonistic effects of the nutrient in soil. So you can see here from figure 1 that in 1950 we have only nitrogen deficiency there. In 1970, after 20 years, nitrogen, iron, zinc deficiency are there. 2010, nitrogen, iron, zinc, boron, molybdenum, etc. So day by day, the multi-nutrient deficiency has been increased, and the main reason is that the farmers are not carried out the soil analysis, and they are applying the fertilizer as per his wish or the advice of the merchant, and they are giving the chemical load into this on the soil. The farmers are believe that when they are apply the more chemical fertilizers, then they will produce the maximum crop production, but it is not true, because the farmers are complaining now that due to excessive chemical fertilizer, our soil become hard and crop productivity has been declined. So I remember that during 1950, 1960, 1 bag of urea in 0.25 hectare land is sufficient for the chemical fertilizers. But at present, in 2022, we should have to apply 2 bags of urea on the same piece of land, that is 0.25 hectare, although the farmers could not get the response as they obtained 1950, 1970. So the reason behind this that the soil become dead. We have killed the microorganism, and you know that the earthworm is the friends of farmer, but we have also killed the earthworm and microorganism. So you know that the role of the microorganism, that when you apply the chemical fertilizer or native nutrients in soil present, they make available to the plants in soluble form. So plant will take the nutrient in soluble form. Then importance of micronutrient agriculture production. Micronutrients are required by plant in lesser quantity, yet essential for the plant growth and survival. Major nutrients required for in larger quantity, secondary nutrients lesser than major nutrient, and micronutrients are required in very, very lesser quantity. But if the deficiency of the particular nutrient observed in a plant system, then the crop yield has been declined. Yield and quality of agricultural product increase with micronutrient application. Therefore, human and animal health is protected with feed of enrichment plant material. Each essential element only when can perform its role in plant nutrition properly, that are other necessary elements are available in balance ratio for plant. So I told that balanced nutrition is very, very important, and we are apply the nutrient balance in soil on the basis of the soil analysis, then plant will take balanced nutrition and there will be more production of the dry matter and we will get higher grain or protein. So the extremely important role of micronutrient. So proper nutrition management is an extremely important precondition for healthy and vigorous crop. In this regard, crop nutrients are one of the burning factor for higher yield. Crop nutrition should be priority in starving to meet global food demand. After all, without essential macro and micronutrient, there would not be any yield. Even though demand for micronutrient are small in quantity, these nutrients directly affect crop growth and development. So we should not have to neglect the micronutrient, although it requires in smaller quantity, but it plays great important role in crop growth and development. The role of micronutrient crop production. So boron, copper, iron, manganese, zinc, boron, molybdenum, and nickel are such micronutrients required in minute amount by plant, but inexorably play an eminent role in plant growth and development. Effective function performed by micronutrient, plant metabolism, reproductive growth, production of carbohydrates, chlorophyll synthesis, nutrient regulation, fruit and trade development. And micronutrient. So in this video, we will see only 4 micronutrients: iron, zinc, manganese, and copper. So micronutrients play a key role in the metabolic activities of the plant. Function of each are given below. Iron in the soil is the fourth abundant element on the earth, but its amount was low or not available for the plant and microorganism needs due to low solubility of mineral containing iron in many places of the world, especially in arid region with alkaline soil. Iron deficiency causes yellowing of leaf. So iron deficiency we are observing arid and alkaline soil because it is not available to the plant. And when we grow the crops and we are supplying the continuous irrigation water, then there will be the chlorosis, iron chlorosis observed in the lake. Then zinc, it helps in photosynthesis and growth regulation. Its deficiency would lead to slower maturity and reduction size of leaf. In the phloem shape, zinc makes up complex with organic acid and low molecular weight and increase its concentration. Zinc deficiency can be seen eroded, calcareous, and weathering acidic soil. Zinc deficiency often accompanied with iron deficiency in calcareous soil. So you know that in calcareous soil, calcium carbonate is more. So the soil pH is higher. So the micronutrients are not available in calcareous soil, although it is present in a sufficient quantity. Then manganese play an important role in photosynthesis and nitrogen metabolism. Delayed maturity and early leaf shape drop are the consequence of manganese deficiency. So you can found the manganese deficiency intervenous chlorosis, and it is responsible for photosynthesis and nitrogen metabolism. So copper is responsible for photosynthesis, grain production, and cell wall strengthening. Copper deficiency results in yellowing of leaves and stunted growth. Then iron is first we see the iron is absorbed by plant root as Fe2, Fe3. So it is active iron. Plant will take the iron in ferrous form. So function of iron, it helps in synthesis of chlorophyll, structure component of porphyrin molecule like cytochromes, hematine, Hems, ferrichrome, and hemoglobin. These substances are involved in oxidation, reaction, respiration, and photosynthesis. Structure component of non-hemine compounds like pyridoxine, constituent of the enzyme system, cytochrome oxidase, catalyze nitrogenase reaction in plants. Deficiency symptoms. Deficiency symptoms occur in younger leaves since iron is immobile element within a plant. So you can observe the deficiency of the iron on top leaves, means young leaves. It occurs in soil of calcareous or alkaline soil and poorly drained waterlogged soil. So you can observe the deficiency of the iron in calcareous or alkaline soil and poorly drained soil. Younger leaves develop interveinal chlorosis with progressive rapidly over the entire leaf. Severe cases, entire leaf turn yellow color. So in this deficiency of the iron, you will find the vein remain green and other portion become chlorotic. So I can show you in deficiency of the iron. So I told that iron deficiency in corn maize. So I told that the vein remain green and other portion become chlorotic. It is a typical characteristic of the iron. Similarly, iron deficiency in rice. You can see here the yellowing belt of the rice, and you can't get any crop production. Then iron chlorosis in sorghum. Then iron deficiency in banana. You can see here. Then iron deficiency in mango. Then iron deficiency in citrus. So I did PhD in citrus acid lime, and I have taken at that time soil application and foliar application, and I am presenting here the control of iron chlorosis. On the basis of my experiment, the results suggested that the spray of 0.1% iron as ferrous sulfate, that is 0.5% ferrous sulfate in 0.05% citric acid solution, proved more effective compared to other treatment in controlling this iron chlorosis. So for that, we have to take 10 liters of water, and we should have to add 50 gram ferrous sulfate and 5 gram citric acid solution in 10 liters. You can also, the treatment is also IS1C treatment having ferrous sulfate 0.25%. So half than the 0.5%. And citric acid 0.025%. Foliar spray can control the chlorosis of these citrus trees. So both treatments are effective for the control of the iron chlorosis. And here you can see the control plot. And here I have sprayed at that time ferrous sulfate 0.5% plus citric acid solution. So you can see the recovery here 0.25. So more recovery in 0.5% as compared to 0.25%. So I have taken the GIF, iron deficiency, the most common nutritional disorder in the world. And iron toxicity in rice crop cultivation under acid sulfate solution. So you can know that the micronutrients except molybdenums are more available in slightly acidic soil. But when the soil pH is 3, 4, then the availability of the iron is greater and it is toxic to the rice crop, particularly the acid sulfate soil. Then let us second nutrient, manganese. So it helps in chlorophyll formation, involved in photosynthesis, particularly in evolution of oxygen, involved in oxidation reduction process in decarboxylation and hydrolysis reaction. It involves in enzyme system and various enzyme reaction in the citric acid solution, cyst cycle. It is a substitute for magnesium in many of the phosphorylating and group transfer reaction, deficiency of manganese. So manganese is immobile in plant deficiencies shift out in the younger leaves. So similarly, iron, you can also observe deficiency of manganese on the top leaves or younger leaves. Interveinal chlorosis occurs, so vein remain green, other portion become chlorotic. So in oat, gray speck, tracks, peas, marsh spot, sugar beet speck yellow, sugar cane, phela blight, midrib, pale green and white. Efficiency increase aspartic acid and decrease glutamine. Increases respiration. And fifth one is accumulation of nitrogen compound mainly as amines. So you can see here the manganese deficiency in corn, maize. So pale grain to yellow discoloration between veins of newer leaves can be confused with the Fe deficiency. Then manganese deficiency symptoms in corn and wheat. You can see here corn and here the wheat. Then diagnosis manganese deficiency in oat forage crops. Then manganese deficiency in cowpea. Then manganese deficiency in mango. Then manganese deficiency in banana. Then diagnosis manganese toxicity in krill peas. Because it is a healthy plant and you can see here toxicity of the manganese. And third micronutrient is zinc. So essential to formation of growth hormones, help in reproduction of certain plant, stabilize ribosomal fractions, influence the activity of dehydrogenase enzymes, involved in oxygen metabolism like tryptophan synthesis. Deficiency symptoms of the zinc, light yellow or white area between the vein of leaves, particularly older leaves. So the zinc deficiency will observe in the older leaves or middle leaves, death of tissue, discolor, malformation of fruit, reduced growth hormone production, cotton white bud little leaf, citrus mottle leaf, potato fern leaf, fruit trees, rosettal abnormal growth, and paddy Khera disease. It is a famous Khera disease. So we can see one by one the deficiency symptoms of the zinc. So you can see the typical deficiency of the zinc in maize. So severe stunting caused by shortening of internode. So here you can see the height of the maize plant, healthy plant. Whereas on left side, it is a zinc deficient plant. So you can see the height. So here there will be the shortening of the internode. So the plant remains stunted. And here the internode length is normal, and we will observe the higher plant height. Normal plant is in right, so it is a normal plant. So you can see here 2 to 3 times, 3 to 4 times greater plant height when you are applying sufficient zinc to the maize crop. Here, when you are not applying the zinc, you are applying nitrogen, phosphorus, potassium, but your soil is deficient in zinc, then your plant will remain stunted and you are getting very less crop production. So here you can see the farmer have applied all the nutrients except zinc, and you can see the growth of the maize crop, corn crop. Then rice, I told that zinc deficiency in rice, Khera disease of rice. So zinc deficiency of rice. In Hindi, it is धान में जिंक की कमी खेरा रोग बोलते हैं. The such type of question has been asked in the undergraduate BSc Agriculture degree program. Then diagnosis zinc deficiency in wheat. So it is deficiency of zinc, so rice and maize and wheat and oats are the susceptible crops for the zinc. Then zinc deficiency in tomato, you can see the growth of the tomato. Then zinc deficiency in BD cotton, you can see here zinc deficiency in soybean. So you can see here zinc is important nutrient for higher crop production. Then visible symptoms of zinc toxicity in reciprocally grafted plant of the zinc. So here the zinc, it is a control plot. and here they have applied zinc nitrate, so there will be toxic effect of zinc. Then last one is copper. So I have given the GIF copper mine, so you are remember what is a copper. So copper is absorbed by plant as cupric ion, Cu2, normal concentration in plant 5 to 20 ppm. Function of copper: 1. Essential for the synthesis of vitamin A, act as a catalyst in respiration, act as an electron carrier in enzyme which bring about oxidation reduction reaction in plant, constitute of chlorophyll. So deficiency of copper, chlorosis, withering and discoloration of terminal bud, dead tissue appear along the tip and edges of leaves, multiple bud formation in the leaf axil, and malformation of leaves, guava, cracking of fruit and terminal bud dieback. So cracking of the fruit of the guava and the deficiency of the copper. Then heavy liming, excessive application of nitrogen P induced copper deficiency. So you can see here copper deficiency in wheat crop. So here you can see the panicle of the wheat. Then copper deficiency in rice crop, copper deficiency in oat crop, copper deficiency in banana crop, copper deficiency symptom tissue culture banana plant. Then tomato nutrient deficiency of copper. You can see the growth of the tomato plant. Then effect of biochar application of copper toxicity in sunflower plant. So when you are apply the biochar, then the ability of the copper has been increased, and you can see the toxicity effect of the sunflower. Then synergetic reaction with micronutrients, avoiding antagonistic interaction and increasing synergistic one can have a positive influence on yield. Possible strategies for this include using fertilizer that combine synergistic nutrient, release antagonist nutrient at different rate, and use different nutrient uptake routes such as foliar and soil application. Other nutrients and micronutrients have also been shown to have synergistic reaction. In one study, the addition of more zinc resulted in a higher yield of wheat in calcareous soil, and the yield increase was largest at the highest addition of macronutrient. So zinc have a synergistic effect, and when you are adding the higher zinc along with the addition of the macronutrient, that is NPK, then the yield has been increased of the wheat crop. Then antagonistic reaction with the nutrient, phosphorus and micronutrients. So the interaction of phosphorus zinc is designated as the P-induced zinc deficiency. Suppose your soil have a sufficient phosphorus in soil. Suppose 100 kg P per hectare. And as per the recommendation of the State Agriculture University, suppose you take the maize crop, then you apply 100 kg nitrogen, 50 kg phosphorus. So farmer has not carried out the soil analysis, and the farmer are applying the 100 kg nitrogen and 50 kg phosphorus to the maize crop. Then what happened? You know that there will be antagonistic effect between phosphorus and zinc in soil. So when you are apply your soil has a sufficient content of the phosphorus, and again you are apply 50 kg phosphorus to single superphosphate or DAP, then whatever the native zinc available in soil has been not taken up by the plant. So in such cases, you are wasting of the 50 kg phosphorus per hectare, and you will lose 15 to 20% crop yield of maize. Therefore, I am again and again requested to the farmer Before sowing of the crop or planting of the crop, we should have to carry out the soil analysis, and as per soil test value, we have to add nutrient, manure, and fertilizer in soil. This nutritional disorder in the plant is commonly associated with high level of available PATE, I told that native PATE, or with application of phosphorus to the soil. This deficiency symptom can be corrected by application of zinc fertilizer. So at that time, you should have to apply zinc fertilizer. Zinc and other micronutrients. The application of zinc has been found to depress content of extractable iron. So when there is zinc is apply higher, then it depresses the iron. But increase Mn in submerged soil and rice plant. Mutual antagonism also between zinc and Mn and zinc and iron was also observed in the rice plant. So the relative mobility of iron and Mn was inversely related to mobility of the zinc. If zinc was applied in greater amounts than recommended, the zinc induced copper deficiency in wheat and barley. Suppose your soil status is medium, then we are recommending to the farmer that you should have to apply 8 to 10 kg zinc sulfate per hectare. But instead of 8 to 10 kg, you are applying 25 kg zinc sulfate per hectare. Then by application of the higher zinc, it induces the copper deficiency. So whatever the native copper present in soil, it will not allow to the wheat and barley crop. So you will observe the deficiency of the copper and you will lose the crop production. Okay. So copper and other micronutrients. High copper in the soil cause iron-chlorosis in the citrus. So when you are apply the higher copper in soil, then there will be a problem of iron-chlorosis. So both have the antagonistic effect. The growth of lettuce at any level of application of copper was affected by the application of iron supply. The toxic effect of copper can be reduced by the application of iron. Suppose your soil have higher copper, and then you want to reduce the toxicity effect of the copper, then you should have to apply ferrous sulfate. So what happens? There will be a reduction of the copper uptake, or reduce the copper uptake, and there will be no any toxic effect in the plant, in the citrus plant or other plants. So I am working at a Shivam Agri Clinic and Laboratory Bhavnagar, Gujarat as a chief soil health specialist, Bhavnagar. So carry out soil, water and plant testing for nutrient as well as plant disease diagnosis, bacteria, fungi, nematode from soil and plant samples at our Shivam Agri Clinic and Agri Laboratory Bhavnagar, Gujarat, India. The website is shivama.in. Another firm is Mati Agromart Private Limited, Bhavnagar. The website is www.matiagromart.com. Please contact helpline number +91 63 59 59 59 59. And we have ICP OES scientific instrument for carried out the soil, plant and water analysis. It is the latest instrument globally available. So it is available at our Shivam Agri Clinic, Agri Laboratory, Bhavnagar. And if you have any interest, then you can see the video of our laboratory. Then correction of micronutrient deficiency. So suppose your soil have zinc, then you have carried out the analysis, soil analysis, then 0.5, then you have to apply 25 kg zinc sulfate per hectare. If your value rated 0.5 to 1, then you have applied 8 or 10 kg per hectare zinc sulfate, having 21% zinc. When the value is greater than 1%, then sufficient. So when you observe the deficiency of the zinc, then you should have to apply 0.5% zinc sulfate and make neutral pH solution with pH buffer solution, or use dilute sodium hydroxide and 2 to 3 spray we require. if you find the deficiency. Similarly, in iron, when there is a less than 5 ppm iron, then you should have to apply 50 kg ferrosulfate per hectare having 19% Fe. Then your value is 5 to 10, then it is medium iron. Apply ferrosulfate 15 kg per hectare. When there is a greater than 10 ppm, then if you will observe the deficiency in standing crop, then foliar spraying of 0.5% ferrosulfate and make neutral pH solution with pH buffer solution or use dilute sodium hydroxide to spray 2 to 3 spray. Similarly for manganese, so for manganese less than 5 ppm low, apply 40 kg manganese sulfate per hectare having 30% Mn. Medium 5 to 10, then you have to apply 12 kg per hectare for manganese sulfate. When there is a high greater than 10, then if you will find a deficiency of the manganese on standing crop, then foliar spraying of 0.5% manganese sulfate and make neutral pH solution with the pH buffer solution or use dilute sodium hydroxide 2 to 3 spray is sufficient. Then copper. So copper less than 0.4 ppm, low copper, application of copper sulfate, 25% copper at the rate of 2 kg per hectare. And the value is 0.4 to 0.6 medium, then you have to apply 0.6 kg copper sulfate per hectare. Then high greater than 0.6, then if you will observe the deficiency of the copper in standing crop, then foliar spraying of 0.5% or 0.4% copper sulfate and make neutral pH solution with pH buffer solution or use dilute sodium hydroxide and 2 dose spraying are required. So thank you very much. And if you have liked my video YouTube, then please do not forget to subscribe. Thank you very much. Now I will I want to make the micronutrient another YouTube on boron, molybdenum, and I will show you, I will present you in a short period.